things from this chapter are going to be covered on that. So this is a good one to understand each of the tests. There's quite a few tests. Okay, so am I going to have to separate you guys in the back? Yeah, why don't you come over here and sit? I mean, because you're sitting there talking.
I didn't even start talking yet.
Pardon me?
I didn't even start talking yet.
Oh, you didn't hear me talk yet? Okay,
No, I didn't register that we're starting a lecture.
Oh, okay, so we're starting a lecture. So in this one, you're going to have a lot of tests that I'm going to go over, and it's really going to be important that you understand each of the tests. So if you don't get something, let me know, because these are the kind that you either want to draw or on the final that we have for the class, they're usually small essay questions where you're given a disease and then you have to use the techniques that you would use to be able to isolate that disease and discover what it is. And a lot of these tests are ones that you'll need to understand. So this is on immunization and immunity, and these are some of the tests that we're going to do. This is called serology, and this means every test that we're going to do is going to be using the serum of the individual. And this is just a kind of a little clicker question to introduce us to vaccinations, which we should already know who created the first vaccination or tried the first vaccination. And who was that? Oh, let me get your, well, I don't have them today. So, but you can kind of, somebody volunteer an answer. Who was it? Yeah, Jenner, exactly. It was Jenner. And so there was two words that are associated with vaccination. One is variolation and one is vaccination. So variolation is where you actually take the organism, the real organism, and that you inject it into someone. And what you try to do is just get a small dose so that they will make antibodies against it. And Jenner didn't do that. What did he use? Cowpox, right. So he injected the little boy with cowpox because he had seen the milkmaids. They got lesions on their hands, but they never actually got sick, and they also didn't get smallpox. So he made that connection. So this is really where the first vaccination actually occurred. So in this particular chapter, there's lots of nomenclature. I'm sorry that there is, but with that comes being able to understand what each of the terms are and how to use them. There's also going to be 4 major mechanisms that we are going to talk about on immunity and 6 vaccination strategies. For those, I really pared it down to just a few things that you have to know about them. So I've, on a couple of slides, I've isolated that. There are also a couple of slides where there is a lot of information about the vaccinations, but you won't have to look at those, and I'll tell you which ones you don't. So we want to know what the role that vaccines play. I think I don't know, not everybody thinks vaccinations are good, but we can probably agree that vaccinations have been a lifesaver on some diseases. Yes?
Nomenclature, so are we going to be expected to be actually able to name them, kind of like how other classes are? Like, how important is nomenclature going to be? Like, memorizing?
So when you say them, what we haven't gotten any nomenclature yet. So
I mean, like in reference to like chemistry courses, how nomenclature we have to actually like look at a molecule and be able to like name them. Is it the same as this?
Well, you'll see when I get to a slide. I'll tell you what you need to know.
Okay.
Yeah. So it's not like chemistry. There's not a molecule that you'll have.
Okay.
It may be a vaccination that you need to know, but there's not that much memorization. Okay, and then lab tests, those are one of the things that are going to be important. So those you almost have to memorize everything about what's going on in the lab test to really understand it and know when to apply it. And then how they can help us make decisions. So these are the kind of highlights that we'll have. Nomenclature only because we talk about serology, so you should know what serology is. We give a good definition here. It's the branch where we're looking for serological reactions. And serological just means the reactions are going to happen in the blood. So serological reactions and epitope-antibody interaction is going to be under laboratory conditions. So this is not we're testing an individual and then we're pulling their blood and looking to see what the test happened. We're going to use their serum, and then we're going to actually do tests with it. So every test that we use is using the patient's serum. And then serum is that fluid part of the blood, and it has minerals and salts and proteins. And what else does it have in there? If we're going to do serological tests, what's got to be in the serum? I think we had this on a couple of lectures ago.
Antibodies?
Antibodies, yes. So the antigen that we're going to get, that is not always going to be in the serum, but the antibodies that your body makes against it will be in the serum. So we'll be able to do a lot of tests to find out, have you been exposed to that antigen before? Are you currently being exposed? We'll be able to look at the types of antibodies that are in the serum. So that's a real important point. So immunity, that's the protection, and this is against a specific disease. So before we had the innate immunity, and remember it was just something that was the first responder whenever you had a pathogen that entered the body. It did a lot of generalized things to get rid of that pathogen, or at least kill the pathogen, or somehow stop the pathogen from entering into the bloodstream. And this now is very specific. So we are using the types of the immune system that is the B cells and the T cells that are very specific for an antigen. It can pass right by another pathogen and not even look at it. It will only look at the things that it's very specific to. So this immunity, using the serum, is using very specific immunity. And so this is going to be for our adaptive, and there's different kinds of things that we're going to talk about. We're going to talk about active immunity and passive immunity. So there's about 3 slides that we repeat this over and over again. So active immunity is when your body is going to respond to something, and then it produces antibodies or T cells against it. So in other words, your body has to see that pathogen. So that makes it then going to be active. And so when it's active, it's going to start producing long-term memory cells, it's going to produce antibodies, etc. However, there's also passive immunity. And this is when your body is going to get some antibodies from someplace else. And a good example could be a snake bite. So you get a snake bite and your body immediately has to neutralize that snake venom in your blood. So you can pull off the shelf at most hospitals, the type of snake that's in the area, they'll have anti-venom. So what they do is they give you IVs of anti-venom. So those IVs are immediately getting into your bloodstream, they're going and they're surrounding that venomous molecule that's in your body, and then it will dispose of it. Your macrophages will come and dispose of it. You can't wait, if you get a snake bite, you can't wait until your own immune system kicks in. It cannot kick in fast enough. And your own innate immune system cannot deal with it fast enough. So one of the things they do is passive is when you receive antibodies. But when you receive antibodies to take care of something like a snake bite, it means that your body is never making antibodies against the snake bite itself. It never had the opportunity to wait for a week to have the macrophages bring around the piece of the snake venom, go to each of the lymph nodes, and then you make antibodies against it. It never has time to do that. So your body never makes its own immunity. And that's what we mean by passive. You get antibodies from something else. Antibodies are proteins. They only last about, a month, maybe up to 4 months, and then they're gone. So if you want to have active immunity, you're going to have to actually either get sick with this, or you're going to have to see it in some way with your own body. Okay, so here's some things on active immunity. This is where you get sick, so this is called natural acquired active immunity. That's another phrase that you need to memorize, and that's part of our active part. Another part of active is an antibody. So this is artificial though, but it's artificial and it comes from a vaccination. So there are vaccinations that we get that give you parts of a pathogen. And so it gives that part to your body in a very small dose. Your body sees that pathogen, at least the part of the pathogen, it's not the real pathogen, and it sees it enough so that it can detect that there's a pathogen in the body and it starts making antibodies against it. Okay, but not all vaccinations allow that to happen. Some of them, you need booster shots because if the bacteria is not multiplying within your body, the body does not sense a threat. They see that there's a particle, they make a couple of antibodies against it, the particle goes away, and then they quit making antibodies. So you usually need to have booster shots to give your body another site of those pathogens, so then it will then make those memory cells and make long-term resistance against whatever disease that you've got your vaccination against. So these are two terms. You got naturally acquired active immunity. That's when you get it yourself. You get sick with whatever it is. You go through all the suffering of getting over it. And then the artificial acquired, and that's when you are given a immunization of a vaccine. And there's several different kinds of vaccines. They each will give you different types of immunity. So these are two things. These are under active. So you have naturally acquired active, and you have artificially acquired active. Okay, make sense? All right. So artificially acquired active, we can talk about that a little bit more. So that's vaccinations, and we're going to talk about this a great deal more. And this really prepares the body for the potentiality of that pathogen getting into the body. A lot of times you get a vaccination way before you ever think you're going to see something, or maybe you want to avoid ever getting sick with a disease, so you get a vaccination. Or maybe you're going overseas and they have something that we never see here in the United States, and so you get a vaccination against that. So these are usually some kind of treated or altered microbe. They're not the real pathogen, or the real pathogen, but they have alterations in them that they are not going to be harmful. So just enough of their epitopes are going to help your body be able to make antibodies against them. So when you see the pathogen, your body sees it, it will be able to use the antibodies that it already knows it has against that pathogen. So the idea is what you want to do is you want to make memory cells, and then you want to make those primary antibodies so that your body's going to be equipped to deal with whatever it is that you get later on. Okay, and because your body is already seeing this pathogen through a way that the pathogen is not infective, if you do see it, you may never know that you've gotten infected. Your body will already have the immunity against it and will be able to fight it off. So you may never recognize whether you get that disease or not. All right, so now there's another kind. So we talked about active. So now you have passive immunity. And this is when the antibodies are already transferred to you. And this is congenital from your mother to the fetus. And this usually is IgG, and it lasts maybe 4 to 6 weeks, and then it's gone. So then the infant has to start making its own antibodies. It has to be exposed to pathogen. It has to get a way to start making its own set of antibodies, because it really can't get it from the mother anymore. And then another way is when you just go to the hospital or something like that, or because you get a snake bite or you get something else. I think Trump went, when he got COVID, he went to the hospital. They gave him a whole bunch of anti-COVID medication, and he came out, a day later or something like that, and ripped off the mask and said, "I'm cured." So that's really not what this is for. It's really to slow down. You get antibodies against, let's say, COVID, and it's not enough for you not to feel COVID, because you want your body to make its own antibodies against COVID. So you get enough so that you don't really get sick to die from COVID, but at least it gets your own immune system the week or so that it needs in order to make its own antibodies. Okay? Because the idea is if you can see a pathogen, you can make your own antibodies, your own long-term memory cells, and then whenever you see it again, you'll be able to fight it off. Okay, so this is actually just getting artificial antibodies against whatever the pathogen is, and this is it being transferred from the mother. So these are passive, and okay, so you can see here, this is natural acquired passive immunity, and this is artificially acquired. So this bag means it's artificial, this means it's the mother giving the actual antibodies. It makes sense. So we have those same terms with passive immunity, and we also have with active immunity, artificial and natural. Okay, so in this artificial, when you're getting this bag of serum, which has antibodies against whatever it is that you need help fighting, that is serum from someone or something else. And when you have serum, remember we have a lot of proteins and different things that are in serum. Some of the proteins could be complement proteins. These are not your complement proteins. They're not made from your body. So therefore, your body may recognize them as something foreign. So when you get this artificial acquired passive immunity, a lot of times you can get what they call serum sickness, because your body will think all of these things that are within that serum that are going into your body are things that they actually need to fight against, some kind of foreign protein or a non-self thing. So remember, complement, hormones, all this kind of stuff. Any kind of protein that you have in your body is always going through the bloodstream. It's usually a pathway that brings proteins from one place to another. Okay, so that's, it's not extremely common, but it is always possible. Okay. All right, so here is kind of a titer that you have. So if you get a passive immunity where you get the antibodies injected, you can see you get a very high dose at the very beginning. And so they're very, very helpful. You can imagine a snake bite. If you have that venom in your bloodstream, you want to have it handled immediately. So you get the passive antibodies, and then you have a really high titer. Here would be a case where you are maybe just trying to fight off the disease yourself. And so here's your initial. We're just doing the lecture first. Okay. So here we have your initial inoculation. Maybe you get a vaccine or something, and your body starts making antibodies. This timeline is probably about a month on this scale, but it could be up to 4 months. This usually the antibodies wane after about either 1 to 4 months. So this timeline is not giving us any specific times, but it is a long period. So here you get a vaccine, and you can see your body is starting to recognize the molecules in the vaccine and is starting to have some kind of response, making some antibodies. And we know that the first ones that are made are which kind? Which are the first kind of antibodies that are made? IgM, yes. And then the IgG are the second one. And it's usually really robust when the IgG comes in, but the first ones are usually IgM. And it is because what does IgM look like? Oh, it's a pentamer. So it's got 5 different arms that are out there trying to gather in all the pathogens that it can. So the IgM very first, and then all of a sudden it goes into your IgG. And the IgM is usually looking for carbohydrate epitopes also. So those really don't last long, so it's not a good epitope to make antibodies against. But IgM does. All right, so it starts to get to a certain point, and then certain vaccines, because they're not live, they're not multiplying over and over again in the body, you'll need to get a booster shot, because by this time your body is thinking there's no threat, and so it's really not making antibodies anymore. And so you give a little booster and then a shot, and then that really shows the body here is the same pathogen again. So now you really need to kick in on the antibodies. So once you do that a couple of times, you get a couple of boosters, that usually gives you almost lifelong immunity against things. Not always, but it can. Once your body starts making antibodies and has long-term memory cells, they usually last a long time, 10, 15, 20 years. Sometimes even the lifetime of the individual. All right, so you can see passive versus active. All right, so here's a thing that kind of makes you distinguish between all of these. So here is artificial acquired active, here's artificial acquired passive, here's naturally acquired passive, and here's naturally acquired active. So what type of immunity comes from actually just getting sick with the pathogen? Naturally, oh, yeah.
Sorry,
Oh, that's a good thing, yeah. Yes, we could do the C. Yes, when you naturally get sick with something, it's got to be natural, and then it's going to be active. when you see that pathogen, your body is going to make the, take the chewed up piece to all your lymph nodes, your B cells, your T cells are going to be activated, and then you will have that probably lifelong. All right, so perfect C is the answer. These are things I put up here for your information only. But sometimes you like to go back and you like to look at these when you maybe are wondering what types of diseases are involved. So here it gives you the types of diseases. What I'm going to show you in another couple of slides is more just general, so you can see what does it look like. Right now, this says contains live attenuated bacteria. You go, oh, what is an attenuated bacteria? So the next couple slides after, there's two of these, and then the next couple slides will actually give you little drawings of what an attenuated looks like. All right, so these first vaccines, these are not really popular anymore. These contain killed whole bacteria. There's always the case that a bacteria is not totally dead. And so then it could come back again and then actually cause disease for an individual. So there are certain vaccines that we still use, but it is not normally used unless you're going to an area that you have to be vaccinated for. So contained live attenuated bacteria. Again, anytime there's a live bacteria, it's also frightful. Although whenever it's live, that means that you're going to get a very strong immune response because it is living within your body and it is multiplying and dividing. So you have plenty of time to see this organism as you're trying to make antibodies, etc. Okay, and then they have subunit ones. Again, these are just a part of the pathogen. So it's maybe just certain proteins of the pathogen. They're not anything that can ever multiply and divide. Again, it's not easy for you to make long-term memory from something like this because it doesn't multiply and divide. Sometimes you have to get several shots. And then here's another subunit one, but this is against other kinds of antigens, and these are toxoids. So this is just for your information only, nothing that you would have to memorize from it. And then this is on viruses. So these are just some things, polio, influenza, some of the things that you've heard about. So again, nothing that you have to memorize off this, right? All right, this is, again, nothing you have to memorize, but you might be curious about what kind of vaccinations are we supposed to get. And maybe this isn't even valid anymore. I don't know what our current government is going to decide is essential for kids. Now, or even for adults. I haven't been bugged. Every time I used to go into the grocery store, they'd always say, oh, have you had your COVID shot yet? Or have you had this shot or that shot and stuff. But I haven't once now for about a month been asked to get a shot for anything. So I don't know, maybe it's not free anymore. I don't know. But anyway, this is just a good list for you if you were curious about what kind of shots kids get. This is probably about 5 years old, this list, so it may be a little different now. Okay, and now these slides I think are more important for you to put more attention to them. So again, we're going to talk about types of vaccine strategies. This is an attenuated live vaccine. Again, this is a live. So that means that there are some difficulties if you are trying to keep bacteria alive. Let's say you're sending something to Africa or to some other countries. You're going to have to make sure that these are refrigerated so that the bacteria don't die. So that's always a problem. And they give a really strong immune response because they are going to be multiplying within the body. Even though they may be weakened, they're still going to multiply. And that means that your body will see this pathogen over and over again. So this is where you're going to get the strongest immune response. It could last lifelong. But it's not all vaccines are like this. And now we can see with mRNA vaccines and some of us, we may just be doing away with this forever. And then another one, so remember this, a good hint for this one, you have to keep it refrigerated. For inactivated vaccines, they're killed, and they're killed either with chemicals, heat, radiation. There's some method that they be able to kill the bacteria and feel that it's pretty comfortably that it's dead. it induces a much weaker response because it can't multiply within the body. So it sees it once when you first get the vaccination, and then after that it starts to wane off. Unless you get boosters. This one you have to get booster shots. This one you have to keep refrigerated, so attenuated live, you have to keep refrigerated, and inactivated, which are killed, you're going to have to get booster shots. And sometimes the booster shots are relatively soon, maybe a month afterward, and then another 6 months afterward, or sometimes there's a year afterward and 4 years afterward. But it is something that you have to get booster shots for. And then these are toxoid vaccines. These are when an organism secretes a toxin, and that toxin gets in the blood system. So there's vaccines for that particular excreted toxin that's in the body. And these are, tetanus is one of them. So this might be what the tetanus bacteria looks like, and this is what it secretes, this tetanus toxin. It's a neurotoxin. The toxin actually looks like this, kind of as it expands, it looks like this. So what they have done for your body to be able to make an antibody against it, they can't really just give you the toxin itself because they don't want the toxin to be in the bloodstream. So what they do is they attach the tetanus toxin to something else. So these are other molecules that they have attached to this. So the toxin itself can't do anything, but now your body can see the toxin. And so once it can see the toxin, it can start making antibodies against it. So this is a safe way to give you a toxin without it being in the bloodstream, being able to do damage. So they attach to something else. Okay, so this is a toxoid vaccine. And then this is a subunit vaccine. There's several different types, but a subunit just means you take a part of the pathogen, and you show that to the immune system. So here might be hepatitis B, and then hepatitis virus. So this is what the virus looks like, but to make a attenuated type of subunit, this is one part. You can see this is really this part right over here. So, and this is the most immunogenic part of that whole thing. So you want to make antibodies, or you want the body to make antibodies against this part right here, and so you give only this subunit of the pathogen to the body. And it can't do anything, it can't do any damage by itself, but you will be able to make antibodies then against the parts of it that are going to be more prominent and more antigenic. Okay, so this is called a subunit, and it's just some epitopes that are, have a strong immune response. And then we have the conjugated. This is somewhat similar to what we have where we're adding different molecules to a toxoid. But this would be our bacteria. And you can see this portion right here, that's the part that's most immunogenic. So it's very hard to give a piece of this without having the whole bacteria, and the bacteria would be pathogenic to you. So they take some of this, and then they surround it with a non-immunogenic molecule. So they take this, and then them together will display the part of the bacteria that you want to make antibodies against. So this will allow this portion to be exposed and yet unable to multiply or divide. It's just a piece of the bacteria. And so this is called a conjugate vaccine. So you just attach it to something else and have the immune system recognize the portion, this portion up here, which is what you want to make your antibody against. And that way then when it sees the whole pathogen come in, you will already have antibodies against that part. Okay, does that make sense? So each strategy of a vaccine has a little bit different strategy. And then the vaccines that we've made lately, this was in maybe 10 or 15 years ago. It was really revolutionary that they made DNA vaccines. People said it would never work. They didn't put a lot of money into it. So at least it was something that had been tried before we got to COVID. So it was an arsenal that we had never used before, but we had, well, they had tried different things, but it was not really successful. Other vaccines were better. So this time they just pulled that out of the kind of the warehouse, and they said, let's try to do an mRNA. This, when they were doing the DNA ones, they were just kind of taking the DNA and they were putting them into little lipid vesicles, and then they would inject the lipid vesicles into an individual, and those lipid vesicles would deliver the DNA to whatever cell. It didn't matter, and then that cell would make whatever is on that DNA. So they'd tried this before, but not very successful. And now we have the mRNA vaccine. So this is mRNA vaccines. mRNA vaccine is, or mRNA, as we know, is the coding for a specific gene. So if you take the epitope of whatever you want to show to the body, and you take it and you make from the gene, you make the mRNA, and then you inject the mRNA into a body, what will happen, the mRNA, just like viruses do, when they inject their mRNA in, ribosomes go, oh, here's some mRNA, and it will start making a protein. So that's exactly what our cells did. It started making the spike protein. When it made the spike protein, they attached onto that spike protein directions to tell the cell to display on the surface. You know, go to the cell surface with this protein. That's where it belongs. So those instructions from the mRNA then just sent all of these spike proteins up to the surface of a cell. And of course, the immune system saw these and said, what is this? They started making antibodies against it. And now when you actually got a virus in the body, the cells were already had antibodies against the spike protein. So this worked out extremely well. I think just having a little bit of background to start with and having so many companies working on it, they could find this mRNA vaccine pretty quickly. And then as this has mutated, because these the spike proteins are very specific because they have to be able to get into a receptor on the cell. As they started mutating a little bit, then we had to alter the vaccine, but that was very easy to do with the mRNA. They just had to have a new sequence, and they could make new vaccines. So it's probably here to stay and probably going to be somewhat quicker to be able to make vaccines with this methodology. Okay, so this is how the mRNA vaccines work. So any questions so far? Kind of makes sense? Okay. So when we're thinking about vaccine safety, I think this is the biggest thing that most people are afraid of. That's why a lot of people don't want to get vaccines, or it could just be a holistic feeling that the body can take care of itself, and I think we probably all agree almost always your body can take care of itself. However, there are things that are just overwhelming, and when your body cannot make its own defense, whether it's through its innate immunity or its adaptive immunity fast enough, there's got to be intervention. Otherwise, the body would not be able to survive. So because you know, there are people who get sick. Every time I get something with COVID, it takes a day for me to get over that COVID vaccine again. In fact, I hesitate to get it now because it seems like it wipes me out for a day or two, and I can't afford that. I got stuff I have to do. But at the very beginning, I was very willing to do that. So now I'm not as willing. I just don't like the after effects of it. So it's not really toxic. It just kind of gives you a little dose of the COVID, and that's not pleasant. So there's things, all of these people are kind of adverse to taking vaccines because of whatever they've heard. And they say, or they've heard that maybe it causes autism, maybe diabetes, asthma, whatever rumors have been, most of it is not documented. And there is this vaccine adverse event reporting system that you can actually look it up, and it'll have any vaccine that's ever been out there, what are some of the reporting from states and nationally also about that vaccine. So it would give you more data than just maybe listening to something on the radio. Anyway, this is one of the things that's a problem because people don't want to get the immunization. Okay, so here's another little clicker question, so we can maybe do the hand signal things. So which of the following is a drawback to the inactivated vaccines? So these are the inactivated ones. So let me see. So that looks like A. Okay. yours is the best. Is A. Yeah. Okay. Yes, this is A. Okay. So this one you got to get the booster shots. So when it's inactivated, it's great because you get to see the epitopes of that pathogen, but it doesn't come around very much, and so your body doesn't really try to fight it off too hard. So a couple of booster shots and stuff like that, then your body knows it's real. So what must be kept refrigerated? It's got to be live, yes. It's got to be live. And then short shelf life contain live pathogens. You don't have to worry about those. Okay, here's another one. Vaccine using an attenuated virus will lead to what's the first word that we have to put into the sentence? It has to have artificial, so we know that. So if it's attenuated, it's going to lead to. So you're thinking of okay, is that a B? Okay, all right. So that looks like a B. So artificial acquired active, it will be active. Okay, so but it is artificial. Anything that you're not suffering with yourself when you get it, that's going to be artificial. Okay, so these kind of terms that you will have, and I will ask you different scenarios. So that will be something that you should at least think about or practice. Okay, here's an anti-venom. Is administered because of a snake bite to the person. What are you getting? Okay, D. So it's going to be artificial because they gave it to you in one of those little bags there. Acquired, and it's passive. It's going to last just as long as you get that drip in. It's going to hopefully negate whatever that is that's attacking you right away. And then over time, probably within a month, all of those antibodies that you are getting are going to be gone. Okay, but by that time, if it is something that you have in your body's had plenty of time then to be able to make some kind of antibodies against it. But this is usually snake venom, and you never see it again, so your body is not going to make antibodies against it. Okay, I think snake handlers have some resistance, but not enough to take care of full load of toxin. All right. So this is nothing that you have to memorize. We'd never ask you about drawing this again, but this is really what happens with why that snake venom is so dangerous. It has something called phospholipase C. The molecule looks something like this, and what happens is it latches on to the plasma membrane of red blood cells. And so when it does that, it starts clipping them off. And when that happens, the red blood cells burst. So basically, you start bleeding to death from the inside out. And so if you don't take care of that almost immediately, and not immediately, within hours, then you'll bleed to death. And so then you'll be squishy and stuff, and the snake can just eat you up and bother the snake. Yeah, so these are really important. But where have we seen phospholipase C before? And I don't expect anyone's probably going to remember it, but you might. It's some organism that wanted to avoid being seen by the outside, and so it get into a cell, and then when the cell put it into the phagolysosome, it chopped up the membrane so that it could just get out. Now it's inside the cell. It made that little actin tail. It pushed it against the side of the membrane. As it did that, it got into the next cell. That next cell pushed around again, and now it's into another cell. The idea Listeria was trying to avoid ever going out into the cytoplasm, because if it did, macrophages would find it and kill it. So it tried, once it got in and got into a cell, it just propels itself from cell to cell to cell with this little actin tail that it does. But in order to get out of the vesicles that it has to have some way to chop through the phospholipids. And so it uses this thing called phospholipase C, or a derivation of it. So, but you don't have to remember that. That was just, it's just a fun fact. Okay, so vaccination benefits the individual as well as the community. I think most of us have probably agreed that that is the case. So it's very important for public health. Here are a couple of diagrams from the book just to reiterate that point. These are two different vaccines. This one is for polio, and you can see when we got an introduction, first when we had vaccines, and then we had another push of the vaccines, and you can see basically it's 0 now. There are some cases that polio has come back, but not in the United States. And then measles, we did the same thing. We really put a big push to try to get the measles eradicated. The first big push made a big difference, but not total difference. Then they made another big push, and now we were really low. But even now, I think in so where's our Texas? I think they have that break of measles, like 20 cases or something. Doesn't seem like a lot, but it's really concerning because one person can spread the measles to 100 people. So it's important that they catch things like that right away. So anyway, this really shows that vaccines can eliminate diseases, and they're mainly diseases that are coming from human to human. Because human beings, you can go around, like with polio, you can go around every country in the world, and you can count every human being, whether you're an infant, or you're an adult, or whatever you are, you can vaccinate them. But if you're talking about, let's say, mice or something that spread a disease, how are you ever going to inoculate all the mice? You'll never do that. So the only diseases we can ever totally get rid of are diseases that go from human to human, that we are the only vector for that disease. And it's very, very expensive to go around and try to eradicate them. It's better just to get people get vaccinations. So, whenever the outbreak occurs. Anyway, this just shows you that it will actually work. So herd immunity is what many people rely on, and herd immunity is it's where a lot of people get vaccinated, and then when they get vaccinated, the unvaccinated person that maybe is getting sick cannot spread it because everybody around them has already been vaccinated. So here's kind of our scenario. So if we look here, this blue part is a person that is not immunized. So these are not immunized. This person is sick, and this person is immunized. So up here, nobody has got a vaccine. There's a couple sick people, and eventually everybody is sick. Everybody next to them, behind them, above them, they're all sick. Yes?
So you know how like the COVID came, there was like a lot of conspiracy about the long-term effects of it, like what happens in 10, right? So like with the flu, we didn't have that same because it's been through time. What are like possible drawbacks of vaccine? Like
mean for COVID?
Like for COVID, everybody's panicking. I know I've heard that a lot.
You know, I don't know if we even know that yet because it's only been 5-8 years.
We don't. What I'm saying, was there any other vaccination that led to something weird happening to people later on?
I think the only, I don't even know if this is weird, but we have vaccinations of organisms that are latent. So you give a tiny bit of the organism to the person, and it multiplies and divides. You don't get sick, but it becomes latent, and then it will come out later on, like shingles and some of those types of things. I don't know if that's. It would be a drawback if it's a terrible case of shingles, but you don't die. But I don't know. There's any maybe vaccinated disease that has long-term effects. I'm sure if I really research it, you could find something. And then,
I was just curious because I know with COVID that was a huge conspiracy.
Oh yeah.
So
yeah. And you will. Conspiracy is really unfounded information, but you really don't know. Nobody knows, and so it leaves these gaps where it's frightening. You know, when you go someplace and you're not really sure, and someone tells you, go down that alley because this is safer. You go down that alley because someone told you, but you don't really know that it's safer. So, when we're taking the vaccines, we're telling you, do this, and you don't know, and so I think it is scary. Yes?
When they're making things like the flu vaccine or other like vaccines for pandemics or epidemics, how long do they have to make them? Is it like a year-long process? Does that be they make it and they have to test it and see the effects of it?
Well, you can see COVID, so we use that as one example. This is something that they didn't have any vaccine for. The flu we have some information for, but this they made in, I'd say like 8 months or something. We went through a lot of that, wonderful time where you didn't have to go to school and you just, you went outside and you could do whatever. I mean, it was really, to me, I loved it. People kept saying, oh gosh, this thing, I hate it. And I said, go ahead and take a walk, go play, go climb a tree or something. You do something. You don't have to stay inside, but I have to stay inside, I'm afraid of,
Yeah.
So, not me. I absolutely loved it. I took so many dogs for a walk and stuff. So when we had to come back to school, it was a hard thing to do, but I did it anyway. So yeah, that was COVID. But the flu is seasonal. So they can go to Europe and they can find out what kind of flu is in Europe right now. And so then they have time to make a vaccine. And when the flu finally gets to us, sometimes they made the right choice. So they got the right strain that came to us, and the vaccine that we use is pretty effective, and sometimes it's not so effective.
And then obviously with the mutations of the virus or disease, so they just keep making vaccines constantly.
Oh yeah, well that's what we do. That's why you have to get supposedly you have to get a flu shot every year. So a lot of people, they always say older people, which I kind of agree. When your immune system is weakened and you get the flu and then you get sicker and sicker and sicker, and then next thing you're in the hospital and that's it. It's worth it to go get a flu shot all the time. But I'm older, but I'm just over it now. Don't make me go and get another shot.
I'm not older, I'm over it.
Yeah, so I don't know. I mean, I do understand why people don't get vaccinated, but people really prone to sickness and stuff. I was first in line for the COVID thing. I was first in line for all the things that the grocery store said, oh, come and do it, it's free, so I did it. And so now after a while, when I come in, I go a different direction instead of going by the pharmacy, because I know they'll nab me for some other brand new shot that they get paid for. That's really, I'm just a money thing for them. They just, another shot, another person, So, oh dear, what did I push? Was it me? So anyway, vaccines, they're great, and I think hopefully all young children get things so they're avoided all kinds of diseases that could harm them. But anyway, so here's our blue people, and you can see that they are going to all get sick, and then we have some that are getting vaccinated, but we still have a lot of people that get sick. And then once more and more people got vaccinated, this is where the herd immunity comes in. So I do wish all people would get vaccinated, and then I wouldn't have to. But I'll jump in. I will be one of the yellow people if it's ever a pandemic or something like that. I would definitely do it. But now it's not nearly as scary. Okay. So anyway, this idea is people depend on this. So some of the ones that for COVID that didn't want to get vaccinated, they were just they didn't get vaccinated, but they would go in the grocery store being sick and stuff like that, and didn't bother them, and it usually didn't bother other people because they were vaccinated. So anyway, herd immunity is something you need to be able to explain. Why is that important? And not maybe draw little people like this, but do an explanation of it. All right, so now this is where the serological reactions and disease diagnosis is. I think we should talk maybe about 10 more minutes and then take a break. And then will only be a few more diseases, the tests. These tests are really important, so I'll go through them not really slowly, but ways so that if you have a question, you can ask. So these are serological reactions and disease diagnosis. That's the whole point of what we've been doing. And ELISA will be at the very end. And there's one thing I also need to do is I need to show the video. I don't know if you watched it already on the website. They have the video about what our experiment is like. But anyway, so these diagnostic tests that we do, we're going to use the serum, and because the serum has antibodies in it, and they also can have the antigen in it. So we use known antibodies. Sometimes you can pull them off the shelf. We already have an antibody against a particular epitope. So we can use a known antibody and then some epitope that we get in the serum. So some pathogen that we see in the serum, and then we don't know what it is, and so we take a series of antibodies out of little bottles, and then we try to see which one does actually match that. And then we can do something that you have the known epitope. You already know what the pathogen is, and you don't know what kind of antibodies the person has. So you can have the known pathogen, and then you can figure out what kind of antibody, or see if the person has antibodies for that disease. Okay, so there's two ways you can do this. So here's one way. here's one way that we make the antibodies themselves. So we talk about antibodies being made, but this is when artificial antibodies are made. And they're not necessarily artificial, but they're artificially given to individuals. We generally, except for Trump, we generally do not take antibodies from another person and give them to another human. In that particular case, Ebola, we have done that when people have gotten sick overseas, like American doctors, and they bring them back to hospitals, and they take people who've gotten Ebola already, and they bleed them, and they get their serum, and then they get the antibodies out, and then they try to help some doctor that's been over there for years trying to help. And so they did the same thing with COVID. They took people who had recovered from COVID, and they got their serum, and they got the antibodies, and they saved them a reserve for people who were very important that we didn't want to die of COVID. All right, so here's how they make them now. So these are antibodies. You have an antigen that's in this little vial. They're going to take it and they're going to inject it into a rabbit. And so, I guess this is a mouse, sorry, it's not a rabbit. So they inject it into the rabbit, and then the first thing the rabbit's going to do then is going to start making antibodies against whatever this is. And when it makes antibodies, where do the antibodies physically come from? Do they come from the B cells? No, what cell do they come from? The plasma cell. So that's what they're doing, is they're having a plasma cell. They're collecting the plasma cells from this rabbit. These plasma cells are the ones that are going to be making the antibodies. They take another rabbit, and this is a rabbit who has cancer. It's myeloma, and its plasma cells are cancerous, and that means they're going to divide and divide and divide and divide, because you don't want to do this if you're only going to make a few antibodies. You want to make antibodies for years and years and years. So something that is cancerous, you can just save it, and you can be making antibodies for a long period of time. So they fuse these two together, and they call it a hyperdoma. So hyperdoma is a cancerous myeloma cell, or really a plasma cell, and a plasma cell that has the antibodies that we want. When they do this, they grow them up, and then in the serum is going to, or in the solution of the experiment, they're going to have all kinds of antibodies. So here are the antibodies that we want. We collect them, and then we can use them in experiments. Okay? So the whole idea of this is we are going to be able to make various antibodies pretty much at will, and then we can be able to make them and help individuals with them. So there's two kinds that we make. An organism will have more than one epitope on it. Just like a flagella on a bacteria, it has a whole bunch of proteins that make up that flagella, and every flagella on every bacteria is going to be slightly different. There'll be many proteins that will be identical, but many proteins will be different specific to that bacteria. So the epitopes are going to be different. And in this one, we made a monoclonal antibody. We made it to only one epitope on that pathogen. And on this one, we had many different epitopes, so we call those polyclonal. So usually you want a monoclonal, that's the most specific, but polyclonal, like if you're using a sandwich ELISAs, you might use that. So for one of them at least. Okay, so anyway, this is what a polyclonal would look like, and this is what a monoclonal would look like. But the idea, we get these from other organisms. All right, so these are the tests that we're going to talk about, and we'll just maybe introduce two tests and then take the break. So these, again, are serological tests, and that means they're all done with a patient's serum. And we won't do all of them. We're not going to do this rapid test. I think we've already kind of hit that in lab. So you do a titration, and this is for you to see how the patient is doing. Maybe it's a patient that's in the hospital, or they come back, a week later or a week after that. And the titration is you take the serum from the individual, and then you use saline to dilute it. So that's what this tube would be to start with. Forget all the color that you see in it right now. So at the very beginning one, I think this is a 1 to 20, and then they go to 1 to 40. So the 1 to 20, they probably put 1 mil in how much to get a 1 to 20? Put 1 mil into
19.
19. That made a 1 to 20. Okay, so the 1 was probably the serum, and the 19 was a saline solution, and now you have a 1 to 20. And then they did now it's a 1 to 40, so then they did a 1 to 1. So they did half of this and half of this, and then half of that and half of that. So that's how they got a 1 to 40, 1 to 80, on and on. So this titering just took the antibodies that were in here and then just titered them out. And then what they did is they put the same amount of antigen in each of the tubes, exactly the same concentration. So this tube has the same concentration, this one, this one, this one, this one, and this one. And some of them don't even show up because you don't have enough antibodies to be able to bind with them. So as the rise in titer, as these colors get further and further down, that would mean that you have more and more antibodies that you're making. So you're fighting the disease. You have the exact same amount of antigen that you had at the beginning every time they do this, but as they do the titers on your new serum, they can see that you're making more and more antibodies, which would mean that it's a good response, that your body is responding to the pathogen and you are being able to fight it. Does that make sense? So this is something they do now, usually especially people who have been in the hospital for a while. Usually they don't call you back to do this kind of test. They just say, how do you feel? And you go, I feel much better. That's it. You know, they don't charge the thing. But this is, so you're looking at the progression of the disease over time. So high antibodies, titer ratio, that indicates that your antibodies are really active. You're still making antibodies. So it's getting higher over here. And getting low, that means that the disease is starting to fade away. So you can kind of see how the disease is going. Okay, so this is just called a titration. So this is one of the tests. And then this one, and we'll take a break right after this one, this is a neutralization test. And this one, I was going to research before I came here today, but I didn't have time to do that. I really can't imagine that this is something that's done. It used to be done in hospitals, but I really can't imagine that they have this many rodents that they can use. So this is testing, let's say, a foodborne illness. And an individual comes in and they're sick and they want to know what it is. So they want to know what kind of antibodies to be able to give it, or antitoxins maybe. So let's say this is botulism. So they take from the individual, they take out of the serum, they take a molecule. And when they do, they inject this serum into a rabbit. And into a mouse again, sorry, mouse. So when they inject it into the mouse, it's obviously going to kill the mouse because this is botulism. It will kill just about any organism. Then they take the same organism that's in, or the same molecule that's in the serum, and then they put what they think it is. They think it's botulism. So they take artificial molecules that they have for botulism, and they pair it with the pathogen that they have from the serum. And they let it sit for a little while so that the antibodies can bind. And you notice the antibodies, if you had to draw this, you would notice that the antibodies are binding when it's variable region. So don't put the tail region. You have to always put the variable region at the epitopes. So the antibodies are binding onto the toxin. Now they injected this into the mouse, and you see that the mouse lives. So this is one indication that this is probably botulism. But you can never do an experiment and not have a negative control. So they took a totally different kind of toxin, and they took the exact same antibodies, the botulism antibodies, and they put it with this other toxin, and of course they don't bind, and they inject the animal with it, and the animal dies. So what they're really showing is it's not necessarily the organism, it's the antibodies. So these antibodies are the things that are actually protecting against the ability for that organism or that protein to be able to do its neurotoxin job. Okay, so questions about this? Again, it's not really prevalent now, but I think there are cases that they still use it. So right now, couldn't we just use an ELISA? Seems like we should have antibodies against this botulism toxin. We could put that down on the, oh, we haven't done it, I'm sorry. Yeah, this is usually we do after after the Eliza. So, okay, so, all right, so let's go ahead and take a break, and then we'll come back. There's probably maybe 8 more slides of tests. I guess you probably already can see that on there. And then a few that we have to go over quickly on the experiment that we're going to do on Thursday. All right, so let's take a 10-minute break. Oh, and then, yeah, I forgot, then we have to take our quiz. Sorry. But that's okay, we did the right thing. Okay, so it looks like 7:16, so we'll say 7:26. And we'll go by the clock. You can probably see it right here. So we'll go by this one right here. I guess I cheated you. It's really 17.
Smart.
Okay. Uh-huh.
About the serum vaccine,
Mm-hmm.
can you put that if you get like a blood fusion?
That's why they're really so careful about they give you the exact same blood, because blood is different than serum.
Yeah.
Yeah,
Yeah.
because they filter out everything.
Yeah, because I was like,
Yeah. Uh-huh.
because I got an allergic reaction.
Oh, really? It might have been, did you get like antibodies or something, or did you just do like a blood transfusion? Or?
Low on my iron.
Yeah.
Just down to so I was like, oh.
Oh my gosh.
Yeah, so then, I like, right after, like,
Oh, yeah, so there was something in there.
yeah, I was like, how did this happen?
Yeah, because they really check blood pretty carefully to make sure that it's the exact match with your blood type, etc. But it could have been something.
Yeah.
Whoa, that is scary.
It was.
Yeah. I mean, almost everything. I'm kind of the thing, unless you really have to have whatever it is, I don't do it. You know, if someone says, oh, you could get, this little surgery and do something, I say, if it's really going to make a big difference, I'm going to risk it. But if it's a little tiny thing, I'm not doing it. Yeah.
They tried suggesting me to get like the newest COVID vaccine at that time.
Oh.
And it was already like it was like a year ago.
Yeah.
And I'm like, what the heck? I'm like, oh,
Yeah.
And I'm like, I was already young. And then I kind of.
Yeah.
Oh, like,
No, yeah, it's, I'm really trusting, I believe in science wholeheartedly, but it's just, you are.
I already got like the basic one. So I'm
Yeah.
like, I feel like it's like, I should get now. Right now.
If you follow the literature, is there really similar pharmacy and how can every other source is saying, yeah, but it's just kind of crazy because yeah, I couldn't use it for more than that because I was but it just really made me think and so I worked extra hard. Yeah. Because like, like I could possibly like, my mom had this thing and I was like, let me just get a bigger picture and I did, and I was like ready to cry. So it was like all my work, I was like,
So Emily,
Yeah, does that change according to your mood? Is that like
there's so many hair stuff.
so good? Which is not worrying. Okay. Baby application. Don't remind me. I will remind you because you're not waiting till next year and you're gonna apply. And I think that's how I'll apply when this class. Yeah, I'm trying to 6. What? No, June 6 you're gonna be setting. Oh, is it end June 6?
No, June 5th. There's a lot of days.
One to the
So
Friday,
June
June
6th.
6.
Probably like 2 weeks. Are we going out on June 6th? No, we're not on June 4th. On June 5th.
Oh, yeah, let's make June 7th. Oh, it's like you did a whole day of recovery.
You just need to stay to like raw. Just like,
Yeah.
cry, that's really funny.
Bro, it won't feel real. And then I'm gonna feel like I have it on my wicked for anxiety every day after that, thinking I'm just gonna get turned in.
Yeah, so honestly, how it goes.
Guys, I wouldn't do this without you guys. I hope you know this. The second I'm probably without you guys, I'll be doing it.
On the swing fence? Oh my god.
I'm like crashing.
So Emily, can you come here a second? Yeah. Huh? Sorry, what did you say? Oh, I want you to raise. Oh, sure, no. So it was after you told me
This one goes
I
like
know,
that.
but then
And
it
then
gives you an
Sunday.
advantage that everybody else is not writing on theirs.
Paragraph kind of.
Right? They're not dumbing out their writing at that point.
2 paragraphs.
Well, no, you're not allowed to start your exam. That's
LeBron James came back from 1 to 3
LeBron
against
James
Steph Curry. So
Yes.
Well,
that's,
like 20
Yeah, and he's like
yeah,
45.
that's. Yeah, okay, we're done.
No, he's great. I'm not being a hater against him. I'm saying that like he's video.
Okay, so let's go ahead and, well, maybe it's not time yet, maybe.
Kobe's not the go, but like statistically wise, I think LeBron might have less than.
No, 3 more minutes, sorry.
Yeah. I mean, mom,
You were also biased.
Okay, so I want to make sure everyone knows you do not write on your exam before we start. Okay, you don't write any information on the back, you don't dump out your mind and stuff like that. You only start when we actually start the exam. It's not really fair to anyone. Pardon?
You're talking about the back page, like where you put your name?
Yeah, yeah,
Oh.
yeah. So you can't put your name and then put maybe all the information that you got just right at the tip of your tongue. So you wait until the exam starts. Yeah, so it may not be something that everybody knows, so I just want to make sure.
So you said the exam at its most would probably be an hour?
The
At its
exam?
most, yeah.
Oh,
At its most.
I always put an hour down, and after I got done with it, I said, this is really a 30-minute exam, but I know I always do like extra time, so I'm saying it's gonna be 45 minutes.
Oh, so
yeah.
it realistically could be 30,
Oh, yeah, yeah.
but you're saying 45 to.
Yeah. There's a little bit more writing, and that's why. If it was just a multiple choice, I could easily guess how much time it is. But for writing, it takes other people longer to think about it, etc. So it could be the 45. I mean, it's not that much, but more than it has been. Okay, so anyway, let's close. So is everybody here? Shall we start, or it's only 2 minutes early, but it's still 2 minutes early. Okay, that's all right. So let's do that.
No, you're still thinking.
Okay, so this is, again, probably somewhat of an outdated test. There seems like there's many things that we can do. Once we learn about the ELISA, we'll see that ELISA could suffice for this. All right, here's something else that's called passive agglutination. This word agglutination just means clumping together. So precipitation might be another word that we would use, but the idea is that things will clump together. And it's very difficult to get 2 very tiny things when they come together, even if they clump together, to make any difference. You just can't see it. So to make it so it's a little bit easier to see, they use these styrofoam, and it's not always styrofoam, it's other kinds of material that they use to put the either antigen on or the antibody on. And this way, then when they come together, there's a bigger mass. And then when they precipitate, they fall out, you can actually physically see them. So agglutination means that you are attaching to the surface of a bead either your antigen or your antibody, not both, just one or the other. And then when they come together, they will have bigger mass, and then they will fall down to the bottom. They'll agglutinate, precipitate. So in this particular one, I kind of divided this. So I made this green. You can see the green right here. This is going to be the antigen, and here's the little bead. This is the bead cut in half, but you can imagine that these antigen is everywhere. It's totally covering 100% of this bead. But because we sliced it in half, it looks like, the side of it has nothing on it. But this would be totally covered with the antigen on it. And then the red is going to be the antibody. So here is an IgM. And so they put these together, and by putting the antigen on the bead, covering the bead, and the antibodies bind the bead, it's going to be heavy, and then it's going to precipitate out. Okay, so this is what they mean by agglutination. You are attaching to the surface either your antigen or your antibody to be able to make the concentration of it much higher, and then when they come together, they'll fall out of solution and give you some kind of a signal. So, and then this one is just the opposite. You can see the red is the antibodies on here, and then the green, the antigen, is on the organism itself, and then the same thing, the antibodies are on the beads. And again, it looks like the antibodies are just sticking off the side of the bead, but the antibodies are 100% covering the bead. So there's a lot of antibodies there. And you notice again, which region of the antibody is attached to the bead. Is it the variable region? No, it's the FC region, or the fixed region. Okay, it's the stem of the antibody. So just make sure when you draw things that you're distinguishing between what's binding. So this, because we want the variable region to be able to bind the antigen, so they put the stem of it on. Okay, so just making visible clumps, and this is all they just call agglutination. Okay, and here's an antibody, an antibody test, but it's on your blood groups. So just kind of a quick review. We have blood type A, B, AB, and O. This is what the red blood cell type would look like. And if you have A on it, let's say A looks like these purple ones. Type B looks with a B with a kind of a triangular shape. AB, we have type AB, and so you have both the A and the B on, and then O has nothing on it. Okay? So if you are trying to determine if you have type A, you can use anti-A to bind to it, and you would see if you had that in your wells. Or if you were looking for anti-B, or looking for blood type B, you could use anti-B. All right? So here's our splotch of blood they have. Took a little petri dish, and they put some serum in it, and then they put the anti-A in it. So, I'm sorry, they put the blood in it. It's not the serum this time. So they put the red blood cells in it, and they put anti-A. And so what did we get? Did the anti-A indicate anything to us in this slide? Is this type A or type B, or what is it? Pardon me?
Type A.
Yeah, it must be type A. Anti-A bounded, you can see it clumping together. It's not, a big visual, but you can see this. And now we took anti-B because we want to make sure. So we take anti-B, and this is what we see, nothing. So by doing this testing the way that we did, and I'm going to skip the next slide. We'll do it and we'll kind of review it. By doing anti-A, and we see that this A matches, and then putting anti-B over here and it doesn't match, that does let us know that this is not anti-B, and obviously it's not anti-A, but what it also lets us know that it's not AB. Because if it was group AB, A would bind, right? So if this was group AB in here instead of just plain A, when you put anti-A in, anti-A would bind group AB. So by doing the test with B and not having B bind, we know it's not AB, because B didn't bind and A did. And also we know it's not O because neither of them would have bound. So doing a test like this, always having your A and B together, can help you rule out whether it's AB also. Or, yeah, whether it was AB. Because if it was AB, you would see A bind and B bind. Make sense? Okay. So here's the iClicker question I would have given you, but I think it takes a little bit too much time to go through the whole thing. So I'll just read it quickly. So yes, there are two bloods. So if we were going to answer this, we would not answer yes, because there are only two blood groups. That's not true. Yes, if the sample reacts with anti-A, then it is anti-A and not anti-B. Well, yeah, that is true, but we don't know for sure. And then it says, yes, if the sample reacts with anti-B, then it is type B and not type A. Okay. And then it says, yes, because anti-A bound, anti-B did not, therefore it can't be O, because it can't bind at all, and it can't be AB, because B didn't bind, and it can't be B, so it has to be A. So I would want you to make that kind of a decision. I don't expect you to know, well, I expect you to know the blood groups, and I expect you to know that anti-A works with anti-A and anti-B does. But also about the determining the anti-AB group. Okay, here's another test. This is a complement test. This is used really quite frequently. It's a very versatile test because you don't have to change too many things in it. This is using complement, and I told you the only thing you had to know about complement, that it was a series of proteins and a big long gene, and then it's all clipped into smaller proteins, and then they can come together and then make a hole in the cell of a bacteria. Okay? And there was something called a membrane attack complex, and that's when the antibody tails were sticking up, and then the complement actually bound to those tails and then made a hole in the bacteria. So this is the complement that we're talking about now. So here's the patient's serum, and what we're trying to do is we're trying to figure out what particular kind of antigen that antibodies would bind. So it's unknown at this time. So the first thing they want to do is they want to get a test that they can determine if the antigen that they have in the patient's serum or the antibodies that are in the patient's serum is going to react with a specific antigen. So they've got to get rid of everything in the patient's serum except the antibodies. So they take this and they do a heat destruction that would get rid of the complement, and that would get rid of anything else they have in there. So now we just have the the patient serum, and now nothing else. Then they go to a guinea pig and they put the guinea pig complement in. So we need to have complement so that we can see if the antibodies are going to bind the antigen that we put in. So you incubate this all together for about 90 minutes or something, and then what happens is the next part of it, we have the antigen that we know we put in, we have the complement, which we know the concentration that we put in, and we have the patient's serum, which will have the patient's antibodies. And in this one, the next thing we do in the exact same tube, we add sheep's red blood cells. And then we add antibodies to the sheep's red blood cells. So we will be able to have antibodies bind. And now we leave everything else in there. We have complement in that came from the guinea pig. We have the antigen that we put in to what we think that the patient has for their antibody. And we've got the sheep's red blood cells, and we have antibodies to the red blood cells. So now what we know happens is we know that the antibodies will bind onto a antigen. So in this case, if the antibodies bind onto the antigen, the complement is going to bind to the tail of the antibodies. Okay? That makes sense? So and we've let this go for about 90 minutes, and so now when we add the red blood cells, this is after the fact, and we add the antibodies to the red blood cells, if the antibody in the original serum did not bind the antigen that we had put in, that we thought would match the antibodies, the antibody would not have bound, and the complement would not be used up with this antibody. It would be available to add to this antibody, which we put in with the red blood cells. If the complement is free, it was not used up here, it would bind to this tail. What it does, it would come off this tail, it would go onto the antigen, and it would lyse that red blood cell. And the whole thing would turn red. So this would mean that we did not have the antigen, we did not have the right antigen that we put in, because the patient's antibodies did not bind it, and because it didn't bind it, we had free complement, and the complement then bound the antibodies that we put in for the sheep's red blood cells, and it lysed the sheep's red blood cells. So this is a very visual test to tell you the antigen that you picked did not match. So you can try another antigen. So you can do this with whatever you want to put in there. Another antigen, you can do it multiple times. So it's very, very versatile. Okay, so does this make sense? It's kind of a little bit of a complicated test, but it's commonly used because it's almost immediate. You know, a couple hours or something like that, you know a result. Okay, need any other explanation, or this makes sense? Is that a yes? Okay. All right, so here's indirect immunofluorescent antibody tests. I keep thinking we've already done the ELISA, but we haven't. So this is going to be similar to an ELISA, but this is kind of only a one-step process. We're going to put down here, this is going to be for syphilis, and these are in spirochetes. But this syphilis, they put this down, so this is from the patient's serum. And then we're going to put antibodies in. And so we have the patient serum over here. Let's say we'll start with here. So you've got the patient serum here, then we put the antibodies in here. And then we're seeing if the patient serum and the antibody binds. And if that's the case, then we're going to do a fluorescent labeled antibody. And so maybe I should draw this really quick since we haven't done an ELISA. So you have the slide, okay, and you put the serum down, and in here we think there's going to be antibodies, and you have the antigen in here. So if the patient has antibodies, it's going to bind like this, okay? And then we're going to put another antibody, and it's got a fluorescent tag on it. That antibody is going to bind this antibody on the tail, and of course it's not going to walk, and it's got a big fluorescent portion on here. It's called fluorescein. So if this antibody bound the antigen, then this secondary antibody can bind the antibody from the patient, and then it will fluoresce. Okay? If it doesn't, if there is no antibody from the patient, we put the serum down here, and you put the patient's antibodies in there, but it didn't match the antigen, then this just would be washed away. So when you put the secondary one in, and with a fluorescent thing on here, it has nothing to bind, and it would wash away. So you would see nothing. Very similar to what we're going to do with our ELISA. So here you can see the antigen coated patient's serum with the antibody. It just didn't match. So this one didn't match, nothing is together, and this one, when we had the fluorescein, it bound the antibody that was bound to the target antigen and fluoresced. So again, a really quick test. And very versatile. You can put any antigen there to test the antibodies, or you can put any antibody there and test the antigens. Does that make sense? So one of the things on this test is it's important the order that you do things. So if you put the fluorescein in first, so this is kind of a question that could be easily asked. All right, so you do this test, and here's the order that you did it in. What result would you expect? So if you put the fluorescein in first before you have the antigen and the antibody from the patient, obviously it has nothing to bind, because the fluorescein antibody is going to bind the tail of another antibody. Make sense? Another thing you could think of is, what if you put the antibody down first? So let's say you put the antibody before the antigen. So if you do that, the antibody could stick, but if it could stick laying down like this on the plate. It could stick with the bottom up like this, so it could bind the antigen, or it could bind with the two variable regions onto the plate, and then if you put an antigen in here, there's no way for the antigen and the antibody to bind. So there's lots of scenarios if you do the steps incorrectly. So you have to figure out what would you see if you did them in a wrong order, perhaps. Okay, does that make sense for this one? This is just kind of indirect. The reason they call it indirect, because what you see is you're not actually seeing the binding of the antibody and the antigen. You're only seeing a signal from a secondary antibody. Your assumption is that it's binding the tail of the antibody that's binding the antigen. So it's indirect. You don't know it yourself. You just, the test tells you that that's what must be happening. Okay. All right, so here's kind of a labeled antibody test. It's going to be really similar to our ELISA. We have a well, and you will have an 8-well strip. It will look something like this. It will already have your antigen coated on it, and then it's going to actually coat the walls also with protein, so that it won't, other things won't bind. Okay, and then we kind of pick this up where we start putting the antibodies from patient A and patient B, and then we're going to put our secondary. So when we put our secondary in, it's going to have some effect on the tip of it. It's going to have some molecule, and then when you actually, it's going to have some enzyme, and when you put a molecule in, that enzyme will break up that molecule and cause a blue color. Okay, so this is almost exactly what we're going to do, and so we'll have this blue color in our well. Okay, and this is like a sandwich one, and I think you read that maybe in the ELISA thing, so I'm not going to deal with this too much. It's just that you have the antibody with its tail down here, and then it's binding the antigen, and then you have another antibody that comes. This is where you might have to use monoclonal and polyclonal antibodies in order to have it work right. And then you have a reporter or an effector molecule, and this is going to tell you that it binds. So again, it's all indirect. You don't really know. You know what you put on the plate, and you know that you got a fluorescence. You wouldn't have gotten fluorescence or some color change unless all the pieces went together. Okay, and this last one, I think this may be where we end up. Yeah, I'm not going to talk about that. So just a couple of slides, and then we'll get into what you're doing tomorrow. So this is actually an ELISA, and this is still very, very commonly done for HIV. So again, this is the beads that we see. What we're trying to do is we're trying to amplify the amount of the epitope that's on there. So here's our HIV, here's our bead that we have. So this is a purchase bead. It has HIV epitopes on them. There's many different ones. So these would be placed on a bead, and they place them on the bead to make the concentration really high. And then we take the antibody. So this one is, here's a positive control. So we take an antibody for HIV that we know we have an antibody that's against the epitopes. Then once that antibody is going to bind, we wash everything away so that no lingering antibodies are there. And then we put in the enzyme coated antibody. So you can see on the top of this antibody, it's going to have some kind of a reporter gene on it. And then when we put the substrate for that enzyme that's on the antibody, it's going to turn color. And then we're going to quench it with something else, and I think we're going to use sulfuric acid to do that, and then it will kind of turn a yellow color. So this would be a positive test. This would be we know that all of our reagents work. You always have a positive test. And then a negative test, we got the beads. You can see that we don't put any of the HIV antibody on there. If there's no antibody, there's nothing that your secondary antibody can bind. Therefore, when you put in the substrate for the enzyme that's on your secondary, there's no secondary, and so you get nothing. So the well will just be blank. Okay? And then here's a serum from the patient. So you do all this, you do a positive and a negative, and the patient at the same time. So that all the reagents you're using are the same, the technique is all the same. There'd be nothing worse than to make a mistake and giving somebody a false positive or a false negative. So again, here's the serum. So the serum is going to have antibodies in it. If the antibodies bind, then we can put our secondary in, and the secondary has a reporter gene on it. And when that gene is given a substrate, it will cleave that substrate, and when it cleaves it, it's going to make kind of a blue color to it, and then you can put a stock reagent in, and then it will, in this case, it's going to turn yellow. Okay, and so that's how we, when we do ours, we're going to use that sulfuric acid too, and it will turn it yellow. Okay, so this is actually a physical ELISA that is often done. It's done in labs like all the time. So any questions about this? It's exactly what we're going to do tomorrow. Okay, and then these are kind of the ideas what the wells will look like, and these are little pictures of them. I think the ones that you have in your text are in the lab manual are just as good, so I won't belabor this, but as long as you understand what this is and what you're going to be seeing. Now these are, you're going to have strips of 8. These are physical wells. They're microtiter plates. They have some with 24 wells, some with 12 wells, some with a whole number of wells. And so this, you can just hold it in your hand, and you could just be using the pipette and filling them with things. But you are going to get a strip. You're going to get one little strip like this of, I think, 8 of them. So it's really flimsy. You hold it and it kind of bends down and stuff. So you have to be really careful you don't spill things out of it. Maybe you have a solid surface behind it. Okay, so that's going to be the ELISA that we're going to do, and I'm not even going to talk about these two things. So I won't even ask you that question. So that is that for what you need to know from this lecture. But I want to go through these for the ELISA lab. I believe you also have them on the computer, yes? Okay, so I'm going to go really quick through it. So this is obviously from another school that they picked up this particular lab from, and they fashioned it after that. So it's called enzyme-linked immunosorbent assay. So you're absorbing the immune molecule on the plate, and you're going to use an enzyme that's going to show you that you have a successful connection. So immunology, only because we're using the immune system of the body, we're looking for the antibodies, and that's an immune organism or immune molecule. And you're going to detect the presence of both the antigen and the antibodies. Okay, and they can use a color change and use these for lots of different things. You can look at those. So this, I'm not going to go through the whole thing. We've kind of talked about HIV. You know, it makes T cells and B cells. And we know that's where we get the plasma cells are where we get our antibodies. So this is how it's going to work. This antigen will already be placed down on the plate for you or on the well for you. And then this greenish color out here is a protein that they filled the well with. And so what it did is it coated every bit of the well. So the well is now coated that any other things cannot now bind to the well. The only thing that can bind is something that's going to bind to the antigen. So anything else you drop in there is not going to bind. It is only going to be something that binds to the antigen. Okay, and so once you do, when you put in the patient's serum, if it has antibodies for HIV, it is going to bind the antigen. And then you will wash this out with PBS. And there's instructions how you wash it like 4 times. And then you're going to put in your secondary. And so this is a secondary one. And you can see it's binding to the tail of the fixed region of an antibody. So it can't bind anywhere else. That's the only place it binds. It's a reporter. It has some kind of a substrate or some kind of an enzyme attached to its fixed region. And then that enzyme, when you put in the substrate for it, it's going to turn a color. Okay, so this is just testing for HIV. Here are the things that you have. You're going to have these 8 well strips. I really like every person to be able to do this. It's not a real expensive test to do, but I don't know how they're going to allocate the little strips. So if the least amount is 2 people do it together, the most amount would be, or the least amount would be 1 person, and the most amount would be 2 people. I mean, it's just, it's really a quick test. You just do some pipetting and that's it. Okay, so there's going to be different colors. You're going to have to, I'm not even going to explain a lot of it now, but when you have your strip, you're going to be putting these in. You need to be extremely careful. You need to pipette exactly what it indicates that you have to pipette exactly in the wells. Because the color that's going to change has to be uniform in particular wells. So some wells you won't have any color, and some wells you will have color. And then your test ones, whether patient A or B gets the color, is really what we're testing for. Anyway, you're going to have a squirt bottle to put the PBS in, so you'll have a bottle like this, and you just squirt it way up to the top. You want to, I don't think there's any way you can squirt anything off. They're pretty well attached. But you kind of want to be gentle. You just want to take it, squirt it right in the bottom part. You just really want to let it drip in there. And then you'll have all your pipettes and your tips. So again, this is what I said. This has already been done for you. This is coated. Your antigen is on the bottom. Here's your little wells that you're going to have. The wells are, the stuff that you put in there is going to be clear. You're not going to really know, so you have to do some kind of indication where the beginning is, where you have well number 1 and 2 and 3 and 4 and 5 and 6 and 7 and 8. Because they're all matching. 1 and 2 are the same, 3 and 4 are the same, 5 and 6 are the same, 7 and 8 are the same. So you'll put one mark to say I start here, and then these are the numbering of my wells. Okay, and of course you're going to be changing pipette tips all the time. Make sure you don't cross-contaminate. And forget about the greens and pinks and stuff like that. And the instructions you'll have when you're doing it. So about 100 microliters, so you know how to set the pipette to 100 microliters. And you're going to, after you get all that done, you'll have to sit about 5 minutes. And then you're rinsing all the time. You're going to be rinsing this in the sink. I told you the little strip is really flexible, so when you hold it like this, it kind of falls down. And you're supposed to try to get all the solution out. The best way to do is fill it up, dump it out, kind of flick it out into the sink, and then put it down on a paper towel and kind of tap it down. So a couple times last semester, people hit it so hard that they just crushed the little wells. So, you flip it over and then just kind of tap them down. I mean, it takes a little bit to get all the solution out. And you're going to do it that 4 times. Okay, and then you're going to put your antibodies in. You're going to be doing all this stuff step by step. And here's the last 3 things. I think, okay, so we have a solution. This is amber. These things, this is tetramethyl benzoate, I believe. I'm not sure about the last B part. But this is toxic. It's going to be in the hood. So you'll get that out of the hood and you'll use that. And then you'll have to be just careful with it from then on. If it gets on your hands or something like that, you want to, well, you'll have gloves on, but if it does get on your arm or some other part, stuff, you'll want to wash it off right away. And then I believe, we didn't do it last year, but I believe I saw the sulfuric acid in the hood. I think we're going to use that. That's a stop solution. It stops the reaction. And then we'll be able to look at our results. So this sulfuric acid, I think we leave it for like 20 minutes or something like that before we actually stop the reaction. And then we'll go to the machine. The machine will read more than one little strip at a time, but I think last year for some reason it worked better when we read one strip, but I'm going to find out if maybe maybe they have it working better now. All right, so that is pretty much it. This is what you expect to see. You, again, on this one, when we have our quiz on all the lab stuff that we've done, or quiz or maybe even on the final, this is a great test to say, what if you did this? What would happen? What would the result look like? So as long as you understand what each step is going to do, then that should be all you need. And so any questions back there that I can help you with? Or yeah, no? Emily, did you have, no, you're okay? And
Yeah.
good. All right, so then a cleanup, and it just tells you how to clean up. They tell you this is a lab manual. It's really important that you do not throw this stuff down the sink. There's a waste container that you put it in. Again, they're really flimsy little wells, and so you have to be careful when you hold them, because you can even if you move your wrist, they'll flip, and so all the solution inside them will come out. And you just want to make sure you don't have all this biohazard stuff in the house. Okay, and then that's it, I think. So again, you have all of this in the lab manual. I think you'll be able to follow it pretty well tomorrow. Again, the lab itself is probably a little more than an hour. There's a couple wait times and stuff that you have. But then the result that you have, you'll be able to fill out your worksheet at the end, I think, of the whole day. So you should be able to get that done within the same period of time. Okay. All right, so any questions about that? All right, so let me get the quizzes passed out again. Let me open this up. The